Welcome guys to the intense process of DNA replication. As you see, this is the DNA structure. Let's give it a close look. The DNA's double helix structure is built out of tiny monomers called nucleotides. Each one of them contains a deoxyribose sugar, a phosphate group, on one of the 4 nitrogenous bases. These nucleotides are linked together by weak hydrogen bonds to form the famous double helix, but it's not any 2 nitrogenous bases that can be paired or linked together. They all fall under the rule of complementary base pairing. Adenine goes with thymine and guanine goes with cytosine. The 5' and 3' refer to the 5th and 3rd carbon atoms in the deoxyribose sugar ring. The phosphate group attached to the 5' end of one nucleotide and the next phosphate group by the 3' end of another nucleotide have the potential to form phosphodiester bonds. DNA is anti-parallel because the strands go in different directions. Here, we go into the 3 steps of DNA replication. First step is unzipping and winding. In this process, the enzyme helicase unzips the complementary base pairs to get the strands ready for follow-up steps. It creates a replication fork in this process. In the next step, an enzyme called RNA primase will add primers to both leading strand and lagging strand. For the leading strand, the primase adds primers at the start of the strand, just to get the polymerase to start. Then it's done its work. But for the lagging strand, since the DNA polymerase can only add nucleotides in the 5' to 3' direction, the lagging strands from the 3' to 5' direction. The RNA primase needs to occasionally add short RNA primers to give the polymerase a starting point to work backwards. This makes up the Okazaki fragments. Then another kind of DNA polymerase comes in and replaces all the RNA primers with DNA chains. Ligase comes to seal the Okazaki fragments together as we enter the 3rd step, joining. Now we've got 2 identical double helix DNA molecules. Both are made up of one original strand, which is the parent strand, and one newly made daughter strand, so they're called DNA semi-conservative. Yep, that's the process of DNA replication. If you watch to the end, you're a legend, and I'll catch you guys in the next time. Peace.